All right, this is our number 3 out of 24. This is the funders panel. And it's really nice that we have funders from not just the US, but also from Canada and from Japan. And some of you are here in person. The Japanese team submitted a video. It's a really tough time for them. It's 5 a.m. in Tokyo right now. So I think it's great, however, to have all of them here. On the panel, and I will share my slides here, we will see different. Yeah. So we have Chris Kinsinger, Amy Cook from CIFAR, and then Takei Kenta and Yusuke Data from JST. And we also have Reed Shabman, who is presenting on the IMAC effort that is running at NIH, and maybe also telling us more about digital twin efforts. And so first up is Chris, and we will run these presentations and then hopefully have time to answer questions and ask new questions. Chris is the Assistant Director for Catalytic Data Resources in the NIH Common Fund, where he helps transform discovery into health through data science. And he is taking on this enormous challenge to bring teams and data sets and methods and algorithms and technologies from 20 plus different Common Fund projects together to be more than the sum of parts. And this is really not easy, and it's just interesting to learn how this can be done and what the challenges are. So Chris, if you would like me to run the slides, I can do this, or you could share your slide if you prefer.
Oh, you can go. I was prepared for that. Or you can run them. I'll just say slide 3.
All right.
Okay. All right. Well, thank you so much, Katy, and hello, everyone. This is such an exciting event to be part of. I was telling my family the Multiscale human event. It sounds pretty big, so I'm honored to be here and kicking off the funders panel. We can go to the next slide. So wanted to start with the NIH Common Fund and what is it? So as Katy mentioned, we fund somewhere around 26, 27 large-scale complex programs that are designed to be transformative, catalytic, goal-driven, synergistic, and novel. And really take an area of science that's kind of beginning, that has a lot of potential, and transform it into something that is really making an impact in health science and healthcare delivery. So that's Common Fund in a nutshell. I'm going to talk about largely one program that I think is really in the midst of achieving those goals, and that's the Human Biomolecular Atlas program. But before I get there, it is holiday season, so I want to start off with a little holiday humor. So the next slide, in the United States, we have something called Elf on a Shelf, but nothing really can prepare you for an axolotl on Aristotle. An axolotl is a type of salamander, which I learned from the person who helped me prepare these slides, Ms. Dina Procaccini. But I want to start off with Aristotle, because if you know anything about him, he goes way back some 2,400 years ago, and he was all about classifying everything you could think of. If you read physics, it's all about classifying all these different objects and what makes up matter, and the nature of a rock is to roll down a hill. And then if you read metaphysics, he's all about classifying things that you find in metaphysics, and then politics, and ethics. It goes on and on. It's just, each according to its kind. The next slide, Aristotle did not have access to the cell. They didn't have microscopes yet. So it really falls to us to classify the cell. There's lots of different types of cell. There's lots of different organisms, lots of different organs in each organism. And so on the next slide, the Human Biomolecular Atlas program, or HubMap, has begun to classify these cells. And you can see a few of those beautiful pictures here. And I'm going to just give a very high-level summary of what HubMap has done in classifying various kinds of cells in the human body, primarily, in the human healthy tissue body. So next slide. There are different ways you could classify a cell. So you could do it by what genes are really active in that cell and are producing copies of themselves through RNA transcripts. And so this is a seekfish picture showing yellow, green, and purple, 3 different genes that are highly expressed in these cells, and which regions, which cells they're highly expressed in. So gene is one way. You could also, on the next slide, classify genes by protein. And so here's an image of a matrix-assisted laser desorption ionization, or MALDI, mass spectrometry method that is coloring different proteins across cells, and you can see which cells are abundant in the green protein or the purple protein or so forth. This is actually this spatial proteomics method here is Nature's method of the year for 2024. So congratulations to all of you spatial proteomicists out there. So you could do gene, you could do protein. One other way that might be the most logical on the next slide is just simply using size and shape of the cell. We can classify things by size and shape, and pathologists have been doing this for a couple hundred years probably, looking, because cancer cells tend to be slightly different shaped than normal healthy cells. And so this is a codex method showing kind of, looking at the size and shape of the cell and classifying that way. So those are three general ways that you can classify a cell. On this next slide, these are a number of methods that have been used within HubMap, and you can press the animation, just advance to the next slide, and we start the animation. So we can start, with the bulk analysis of tissue and identify all the genes and proteins and RNA transcripts in cells. We can go to spatial proteomics, as you saw. We can also do spatial transcriptomics, which is looking at the gene expression in various cells. There's also lots of different imaging, mass spectrometry, other methods of imaging to look at the cells, look at the various levels of tissue. And then we can get down to single cell sequencing on the right, and all of the different methods that are associated there. So all of these methods have gone on some of the same tissue samples to get a feel for what's the cellular makeup of various tissues. And then on the next slide, we're beginning to combine all of this together, starting with various cell types, and then forming those cell types into neighborhoods of cells, and then those neighborhoods join together to form communities of cells. And then eventually you get up into these tissue units, and the tissue units can combine to form tissues, which of course tissues combine to form organs. So we're really trying to bring in different levels of information for the going from the cell up to the organ. And we've done this across a number of organs within the HubMap program. And I think that brings us to the next slide. Okay, and this spatial transcriptomics, spatial proteomics approach has really exploded in the past few years. HubMap isn't the only game in town. There are lots of different programs represented on this slide that are looking at particular organs like kidney or urinary tract, or looking at particular disease states, like the Humor Tumor Atlas Network is doing the same thing as HubMap, but on cancerous tissue and classifying the cells there. So we've really just seen an explosion of these kind of projects really around the world, and it's a pretty exciting time to be classifying cells and making maps and organ maps out of these methods. Okay, but that brings us up to Ernest Rutherford on the next slide, who said, or at least it's attributed to him, that all science is either physics or philately. Philately, of course, being a fancy word for stamp collecting. So within HubMap and these spatial omics programs, are we merely just collecting stamps, and we're not really getting into the principle of how what we find in the cell is connecting to disease and health? So that's what I'd like to show next, is what we're doing to try to make sense of some of this. So what good is it doing all of these things? Some questions that we're asking now: how do we integrate these data together? How do we relate cell maps to disease? And ultimately, how do we turn these data into real, useful, tangible knowledge? So I'm quickly going to go through 3 different ways we're starting to do this. So on the next slide, this is one way: data integration. You can get a lot of scientists in a lab and try to make sense of this and run experiments and knock out mouse models and cell lines and get a lot of microscopes. That's one way. But as you saw, there's a lot of methods in HubMap that are going into each sample, and each tissue has, thousands, millions of cells. So there's just a lot of information. It's really overwhelming for one human mind, or even a team of very bright human minds. And so we're trying to find ways to use computers to scale so that we can actually handle this level of information. So on the next slide, many of you have probably heard about artificial intelligence. So what can we do if we apply artificial intelligence to this spatial omics field. And so Trey Eidecker, part of the Bridge to AI program, is doing just this, where he's actually generating data, a lot of protein-protein interaction data, and then feeding that into an AI model to try to make sense of functional genomics. How do genes relate to function, relate to the architecture of a cell, and then follow that architecture? How does it change over time as it goes through the cell cycle? So this is one way to do this. And so, so it's using AI to make sense of these data, find out how genes and RNA and proteins and of course, those images relate to the structure of a cell and classify it that way. And ultimately, you could move toward relating it to disease if you're working with cell lines that have particular, usually cancers associated with them. Okay, and then the last slide, I think, is another way, which is called a knowledge graph. So a knowledge graph connects concepts together through some kind of relationship. So we might have a gene as a concept, and maybe breast cancer is another concept. And then you could say that this particular gene, like the BRCA1, is associated with this breast cancer. And so there could be lots of associations like that. And so we can map all of those together in what we call a knowledge graph. And then that's a way of kind of building a structure of the knowledge that we're deriving from primary data. So this particular knowledge graph relates gene associations from pediatric genomics and pediatric cancer and structural birth defects, along with gene-drug relationships, which we got from the LINKS program. And combining those two data sets together, now we have a knowledge graph that predicts drug toxicity for people who are pregnant, and what drugs might cross the placental barrier and could harm the fetus. So maybe you wouldn't want to take those drugs. So it's not a clinical tool, but hypothesis driving. So that's one application of knowledge graphs. You could imagine doing the same thing for spatial omics and making associations between disease and cell type, or some combination of cell types that are associated with a particular disease state for that tissue. So I think that's what I want to say. I wanted to acknowledge the people that helped me put this together on the next slide. So certainly Dina Procaccini, Richard Conroy, Trey Eidecker, the HubMap Consortium, as well as the Common Fund Data Ecosystem, which I didn't talk about specifically, but they're really behind a lot of the knowledge graph work. So thank you so much, and happy holidays.
Thank you, Chris. And I like that you flavored it in the spirit of Christmas. And I think here's more information on the NIH Common Fund. If you want to learn more, please go to these links. All right. Many questions and comments, but I will hold those, and we will have Amy Cook present next. And she's from CIFAR, a Canadian institution that really is responsible for financially supporting this event. And Amy, thank you so much to you and to CIFAR and to all involved.
It's a privilege to be here. Thank you very much for the invitation. And apologies, I'm in a room right now with a cat outside who really wants to get in. So if you hear him meowing, that's
Thank you for making that all work. Would you like to run your own slides, or should I run them?
No, you can go ahead.
Okay.
So again, my name is Amy Cook. I'm the head of research operations at CIFAR. And again, it's a pleasure to be here. You can go to the next slide, Katy. For those not familiar with CIFAR, we are a global research organization that's based in Canada. And really what we do is aim to mobilize the world's most brilliant people from across disciplines, geographic boundaries, and across career stages to drive transformative knowledge in order to solve humanity's most complex problems. We are grateful for funding from our government of Canada, but also from provinces of Alberta and Quebec, but also receive funding from Canadian and international foundations, individuals, corporations, and partner organizations. Next slide. To give you a flavor of the types of initiatives that CIFAR engages in, I'll just highlight what is outlined in our new strategic plan that was launched recently. Again, we really are aiming to drive transformative knowledge and impact at scale. That is our pinnacle aspiration. And to do so, we engage in a number of initiatives to drive early career research development, particularly through our Global Scholars Program, which I'll touch on later. We also have a number of research programs that make up a portfolio of impact clusters, which I also highlighted in a bit. We also engage in a number of activities around foresight. This is a new initiative for us, and it's really to drive an understanding of what are those key issues that are on the horizon where there may not be a lot of funding support behind, and where a non-traditional funder such as CIFAR could really have a lot of value in that space. I'll just give an example. You probably all know that Jeff Hinton just won the Nobel Prize as the grandfather of AI. CIFAR was the earliest supporter of that work by really believing in his vision of what the power of understanding the brain could be to driving artificial intelligence. And so that is an example of us investing in issues on the horizon. We also drive talent identification activity to really diversify our network across the globe. We engage in strategic partnerships also in key strategic regions. And all of this is really to drive the greatest impact that we can in the research ecosystem. Next slide. As I mentioned, we have a number of activity that's centered around impact clusters. For the purpose of this particular event, I think the most relevant are decoding complex brains and data, exploring emerging technologies, and shaping the future of human health. And what we try to do is engage our research programs not only as a program individually, but also across programs, really to drive understanding of how we can further drive impact within these cluster areas. Next slide. For all of those impact clusters, they are made up of a number of research programs. We have 15 research programs in our portfolio, but key to this event is our CIFAR Macmillan Multiscale Human Program. This is a program that is in its very early stages with a goal of integrating data from across spatial and temporal scales across the body, all while considering the diversity of the population, and really to create an unprecedented map of the human body in order to drive medical advances. So a really bold, ambitious goal, but we are very fortunate to have the leadership, which I'll show you on the next slide. The leadership of Gary Bader, who is based at the University of Toronto, Katy, who is on the panel here, from Indiana University, and Sarah Teichman from the Wellcome Sanger Institute. And these three scientific leaders of the program are really meant to drive that vision forward. And to do so, they've developed a program made up of a very multidisciplinary and international group of experts. Next slide. And which is core to CIFAR, is engaging across disciplines, really to drive a very interdisciplinary approach to a complex problem. And so this program incorporates scientists and experts from various disciplinary areas such as imaging, clinical science, genomics, bioengineering, data visualization and user interface design, cell and developmental biology, and computational biology, theory and biophysics. So really a unique composition. And again, it's really to drive a new understanding of this type of question from very different viewpoints. And our goal at CIFAR is to bring these different perspectives together to really try to think about these issues in new and complex ways. Next slide. So as I mentioned, for our research programs, it's really to bring interdisciplinary researchers together and to drive transformative knowledge. How we do that, like we do with our Multiscale Human Program, is to bring together a group of 15 to 25 individuals from around the world and across career stages. And we bring them together 2 to 3 times per year. And that's really to drive a deeply collaborative network that we fund for a 5-year term. And these networks are renewable through a competitive review process, but we do aim to have our programs think about a 10-year plus horizon. And that's really because the problem is very complex. And as we bring different disciplines together, it does take time to build that common shared language and to really start to gel as a group. And so the way that we also build this group's cohesion is around encouraging collaborative efforts. Catalyst funds is one mechanism by which we do that, and those are small seed funding opportunities for program members when they meet and come up with novel questions and ideas, is to really be able to act on those in a very quick and nimble way. And the Catalyst funds is the mechanism by which to be able to act on those ideas that a typical funder may not be able to. And to provide the foundation for potentially larger grants. Next slide. Part of this program and our other CIFAR programs includes the integration of younger early career researchers. These are part of our CIFAR Azrieli Global Scholars Program. This program is really meant to develop highly talented early career researchers into influential research leaders and to shape the direction of very forward-thinking research areas. There's 3 core areas of this program. The first is participating in a CIFAR research program. They are embedded within a CIFAR research program for a period of 2 years. And during that time, also have an opportunity to engage on an annual basis with other early career researchers from different programs that we fund, and that's really to strengthen their leadership and communication skills. And we also provide training to increase their impact within and outside academia as well. And we are very fortunate in this program to have 3 global scholars, Bianca Dumitrescu, Anne Gregory, and Tobias Wenzel. Next slide. And as I mentioned off the top, the work of CIFAR is not without support from a number of important scientific partners. They are listed on this slide, and there's many that we are also engaging with in other regions of the world. And hopefully this you'll see this list grow, but it's an important element of the work that we do to be able to drive this impact forward. So that's all I had to say for today, just to give you a flavor of the work of CIFAR and how it connects into the 24-hour multi-scale human event, and happy to answer questions at the end.
Thank you, Amy, and thank you also for leading our team and making it successful. It was nice to meet you in Tokyo, where we had our most recent meeting, and to just see you in action. And it's wonderful to also see CIFAR embrace forecasting exercise. I think there's much to be gained from trying to predict desirable futures and then also trying to implement them ultimately. And it's good to do this in a data-driven way, and there's now much data available. All right, the next presentation is by a team from Japan. The Japanese Science and Technology Agency actually attended our CIFAR meeting in the Miraikan Science Museum 2 weeks ago. JST is a national agency that plays a central role in Japan's science and technology basic plan, and they provided a video of their presentation, and I think Nicole is going to run this for us. Thank you, Nicole.
Hello everyone, I am Kenta Takei from JST. It is a great honor to present at this exciting event. Together with my colleague Mr. Yusuke, we will provide a brief introduction of JST and our initiatives related to the Multiscale Human Program. JST is Japan's national research development agency that connects a wide range of stakeholders to drive innovation and create new value for our global society. Our stakeholders include academia, industry, and beyond. By implementing Japan's national R&D strategy through research funding, we aim to achieve strategic goals that promote science, technology, and innovation. JST's core functions are these 4 key areas: research funding, public engagement, R&D strategy planning, and information platform and database services. Over 3/4 of our budget is allocated to research funding, showing its central importance to our mission. Our research programs are mainly categorized into 3 types: strategic basic research, international collaboration, and industry academia collaboration. And here are some examples of programs under each category. JST's initiatives cover a diverse range of research topics, each aligned with Japan's STI strategy. Today, we would like to introduce one research area within JST's CREST and PRESTO programs that align with Multiscale Human Program. This area is called UCell. Here I will hand over to Mr. Yusuke, who is in charge of the research area at JST.
From now, I will introduce about the USL research area. USL is a research area that spans across two programs of JST, PLEST and Presto. The aim of Presto and Presto is to fulfill the strategic objectives set by Japanese government. JST specifies research areas to achieve these objectives, such as USL, and appoint a research supervisor to lead the area. The research supervisor then creates a virtual network-based research institute to fulfill the objective of the area. USL aims to catalyze the synergy between team-based and individual-based research. The research supervisor of USL is Professor Atsushi Miyawaki from Riken. The deputy research supervisor Presto is Takashi Yamamoto from Hiroshima University. The concept of USL, Saibo Asobu in Japanese, embodies playful science. The U in USL represents for you, reflecting Professor Miyawaki's vision. In addition, it is a word play on the Japanese kanji for play, which is announced as you. Professor Miyawaki hopes that USL researchers will approach science with a playful spirit and enabling them to embrace and take joy in conflicting the mysteries of science, and while pursuing the unknown with a sense of adventure. The strategic objectives of USL is to develop innovative cell manipulation technologies and elucidation of cell regulatory mechanisms. USL is distinct in that it covers all living organisms. Here are some examples of research themes covered by USL, and I will introduce two projects in the next slide. I would like to first introduce the project of research director Professor Nozomu Yachie. His project is on the DNA event recording cell, which high-resolution molecular and cellular information from individual cells within a multicellular organism can be progressively stored on synthetic DNA tapes. Next, I would like to introduce the project of manipulating parasites by research director Professor Kumiko Tsukui. When conducting parasites manipulation research, Professor Tsukui approaches the parasite with profound respect, viewing them not merely as test subjects but as master navigators of the human body. She tried to discover and develop a manipulate technique to render 5 types of parasites non-pathogenic. Developing and manipulating non-pathogenic parasites unlocks potential applications in improving disease treatment, vaccine, and drug delivery systems. And we focus on initiative to foster human connections. Once a year, we offer a 1-hour free talk program designed to create spontaneous encounters for researchers similar to CIFAR. And the Crest of User initiated 7 projects in 2023 and 6 projects in 2024, as listed below. Crest of User initiated 60 projects in 2024 as listed below.
Okay, thank you, Yusuke. Lastly, I'd like to take this opportunity to introduce JST's international cooperation activities. At JST, we promote international collaboration based on 3 pillars: research funding programs, talent circulation, and international dialogue with partners. Today, I will focus on the Aspire program, which targets research talent mobility between Japan and STI advanced countries. The Aspire aims to stimulate international brain circulation by encouraging researchers to engage in global networks of cutting-edge research. It helps create a virtuous cycle of international talent mobility and collaboration. And here are some key details about Aspire. It focuses on 7 areas set by the Japanese Cabinet Office, and we partner with STI advanced countries to promote talent mobility. A significant portion, 70% of the research funding, must be allocated to research mobility initiatives such as researcher exchanges. The Aspire support scheme works as follows. Through alignment calls, JST funds Japanese research teams collaborating with international partners. These collaborations allow both Japanese and international researchers to use Aspire funds for reciprocal talent exchanges. While alignment calls are open to collaborations with a wide range of countries, JST also coordinates joint calls with selected partners. The next alignment call is scheduled for March 2025. So if you have collaborations in Japan, please reach out to them to apply for this program. And here is the link to the UCell and Aspire program websites. And I will pause 5 seconds here for you to scan the QR code. Okay, that concludes our presentation. Thank you so much.
Thank you very much, Nicole, for sharing. So it's really exciting to see more and more cross-country collaboration and funding opportunities. Oftentimes, it's impossible for one country, even if it is as US, to map the human body at single cell resolution. It takes much more data, much more expertise, much more money than one country might have. And so having these international collaboration opportunities that CIFAR supports, or the Aspire program, which we just saw, supports, is really critical. Before we go into the discussion, it's my great pleasure to introduce Reid Shabman. He was introduced to me by Grace Pang. Grace has been coming to modeling science events that we have been organizing in Washington, D.C. And so we connected via that line of research. But Grace is also working on the National Institute of Biomedical Imaging and Bioengineering within NIH. And Reid Shabman is part of the Interagency Modeling and Analysis Group, which was super interesting to me because they are trying to get together experts on modeling the human body across scales. And this has been going on since quite a while. So way before CIFAR program on the multiscale human came into existence. Maybe together actually with Peter Hunter's work, which was presented at noon today. He is doing fascinating work on multiscale modeling. And so Reid is the acting director of the Office of Data Science and Emerging Technologies, great office to have. And he coordinates the development and implementation of NIAID's data science strategy across its entire global portfolio of research and training programs. So it's a true pleasure, Reid, to have you here, and I'm looking forward to your presentation. I wonder if I should share my screen or if you would like to share your slides.
Thank you for the kind introduction. You can share your screen and I can just say next slide. That'll work.
All right. You should see it
Thank
now.
you. Yes, I can see it. And I really enjoyed the other presentations in this session. I don't have as good of a theme as Chris, but what I'll do to try to get some interaction is maybe I'll drop some links in the chat for everybody as I'm going through, so you can start to navigate around. So it's a privilege to be here today. I was asked by Grace, who's on the top right of your slide here, to come speak to you about the Interagency Modeling and Analysis Group, and I'll try to do that justice. What you can see on the right are 3 images. Grace is a huge fan of taking screenshots during Zoom, and somehow she captured me during a moment at the office. And then Liz Kinetzi is another co-organizer of this group who is at NICCH. So next slide, please. Okay, so what is the Interagency Modeling and Analysis Group if folks haven't heard of it already? So I'm going to just kind of I'm going to drop the link actually into the chat while I give a little bit of background. Hopefully that'll work for folks here. So stay here, listen to me but you can go back to it later. But essentially, this is a group that was started in 2003. A long time before I arrived in around 2020 during the pandemic. And it started from a working group comprised of program staff from 9 institutes at the National Institutes of Health and 3 directorates from National Science Foundation. And I'd say that NSF and NIH are kind of the biggest contributors to this, but IMAG now represents over 100 program staff from multiple government agencies in the United States. And since its creation, this group has convened monthly through virtual meetings and at various locations of IMAG participants. They're all involved in managing. All of the IMAG participants are government officials that manage research programs in biomedical, biological, and behavioral systems that require the development of new and novel modeling analysis and methods. And this is somewhat of a labor of love, Grace would say. This is not, it's a lot of crowdsourced, even from the government side. And so the purpose of IMAG is to provide an open forum for communication among government representatives to share updates to individual programs among various government agencies, so there's no duplication and of efforts. And on the other side, there's folks in the research community doing the real work, which the multiscale modeling members consortium, and they also are involved. And so if you go to the website, you'll be able to see a lot of the activities that are going on. I'll just drop into the link chat here. If you're looking, my goal here today is to try to connect folks. So this is a list of folks that are on the government side that have sort of self-identified as being part of IMAG. They have their email addresses. You can go bother them. And then I think I just closed it, but I'll just hopefully you guys can navigate around and find. There's a number of folks like Peter Hunter, for example, who's been involved in this from the very beginning, and others that are there as well. Next slide, please. And so I think I'll just make the point here that we hope that this effort is greater than the sum of its parts, and that you come join us. You can go onto our IMAG wiki, which I've dropped in the chat, and sign up. And so like, what are some of the ongoing activities? I'll get to those in a minute. But one example of things that have happened, I don't have a slide for this, but I'll drop it into the chat as well, is our working groups. So this is a multiscale modeling and viral pandemics working group that's very active. So the reason I dropped it in the chat is because it's a great group of folks to connect with and hear about what they're doing. They bring in guest speakers monthly, and it's beyond viruses. They started at COVID in 2020. We were all sort of sheltered in place, and it's beyond. It's bacteria, it's cellular, it's imaging. It's all over the map in terms of multi-scale modeling. Next slide, please. And this is just to say that this is Grace's slide, that this started in 2003, but we really try to remain committed to thinking about what is often not thought about at NIH study sections, which is bridging models across scales, all the way from the molecular cellular level up to the organismal level, up to the population level. Next slide, please. And I'd just like to highlight that these are a couple of highlights of what the group has accomplished in the last several years. So I'm just pointing out a couple of works that have come kind of in collaboration with this group. So Ellen Kuhl is a driver of the top paper, integrating machine learning and multi-scale modeling, perspectives, challenges, and opportunities in biological, biomedical, and behavioral sciences. So I encourage you to check that out. And that was also really part of a theme of one of the workshops that this group held in 2019. And the other nice thing about the wiki is that all of these informations are documented, so you can go in and you can say, okay, what happened at this meeting? You can see all the top presentations, you can see all of the speakers, you can see everything. It's all publicly available. And just to highlight another paper that's useful to provide a little bit of perspective about multi-scale modeling meets machine learning. What can we learn? The idea of really the importance of these advanced technologies, such as machine learning and artificial intelligence, really being critical in bridging scales when you're trying to build models. Next slide, please. And so that's kind of what's happened in the past. I'm going to now pivot to a couple slides of what has happened in this group over the past year. So in 2023, I guess, a year ago tomorrow, a report was put out on the National Academies entitled Foundational Research Gaps and Future Directions for Digital Twins. Next slide, please. This was very exciting to our community because this document provided a roadmap and sort of laid some sort of ground rules for what do we mean by something like digital twin. So the definition in the NASEM report is: a digital twin is a set of virtual information constructs that mimics the structure, context, and behavior of a natural, engineered, or social system, or systems of systems, is dynamically updated with data from its physical twin, has predictive capability, and informs decisions that realize value. The bidirectional interaction between the virtual and the physical is central to the digital twin. So this is an aspirational idea. That's a long definition. The point being that perhaps moving forward, we have digital twins in manufacturing and other areas, but translation to biomedicine is often difficult. But the potential for having something that would provide information about how an individual would respond to a drug treatment or a therapeutic or a vaccine in a digital environment prior to the intervention might be useful. That's one use case. Next slide, please. Okay, and so this was the reason that report was interesting enough for the 2024 IMAG MSM consortium meeting to be focused specifically on this topic. And so what I have here, and this is all available through the links I've provided, is a summary of what this meeting was about. So it was in September 30th through October 2nd. And as I mentioned before, all presentations, notes, and recordings are posted on this IMAG wiki. And there was 3 days. It was an intense meeting. The first day was really around defining biomedical digital twins. The focus here, we scoped this to biomedical digital twins, really to understand the nascent digital twin components, and also to identify unique features for digital twins in the biomedical domain. So what's this report telling us about how to build such a digital twin, and what are unique features that we need? Day 2 was really focused on approaches to address biomedical digital twins, or BDT challenges. There are unique challenges for developing a biomedical digital twin. I think I alluded to that on the previous slide, which might differ from other types of digital twins. And there was an opportunity to discuss the needs with experts and compile component resources. And day 3 was operational team science. So one central theme of this was this type of aspirational idea isn't going to be accomplished in silos, but we really need to bridge disciplines and bring people together at the beginning. And so the goal 1 here was to form BDT idea teams guided by team science approaches, and then to present and review reliable, fit-for-purpose digital twin ideas. And I'm not going to go into all the specifics here because the ideas that were sort of brought up are available. But it was really important to think about everything from just how we structure data to all the way up to the regulatory and ethical concerns that might be associated with these ideas. Next slide, please. So it was a little bit of a whirlwind, but I wanted to just, if you take anything away from my talk, it's just to please, go to the wiki, connect with folks, ask us some follow-up questions, get this on your radar. I'll just say 2 slides about my day job. So thank you, Katy, for introducing me earlier. I work at the National Institutes of Allergy and Infectious Diseases and in the Office of Data Science within our office. My colleague Chris is on the call. The NIH is a very complex place. You can think of it as 27 institutes or centers. NIAID is one of those. We have an Office of Data Science, so I represent data science interests for NIAID while trying to work across the NIH. Our mission is to, on the top left, is to expand the breadth, depth, and knowledge in all areas of infectious, immunologic, and allergic diseases. And our dual mandate is also to rapidly respond to new and emerging threats. And I've overlaid our office's mission below, which is to facilitate data use and computational methods in research mission areas, and also to coordinate NIAID's data science strategy across its global portfolio of research and training initiatives. Next slide, please. And so one of the things that we do in our office that kind of ties back to what Chris alluded to and others have alluded to is really focusing on finding data. So finding data, if you're going to build a multi-scale model, finding data is a critical first step. This is one effort that we've worked on in our office, which is like a PubMed for data. It's a metadata catalog. It allows you to return search results from different repositories all at once. We try to focus on more immunological and infectious disease data sets. You can go into the portal. There's a website called Kayak, where maybe you're going on vacation and you want to look for your rental car and your hotel and your airplane. You don't go to each of those individual sites, you go to Kayak, and that's kind of the idea here. So maybe you type in tuberculosis or whatever the question, or an immune disease, whatever you're interested in, and it's going to return results from 35 repositories at the data set level, an additional 20 resource catalogs. So basically, if you type in tuberculosis, TB portals as a resource might come up. We don't have that at the data set level, but we'll alert you to the fact that, hey, maybe you didn't know about TB portals, and it can take you there. You can work to get access. We also have a search enable for computational tools, so you can start to find computational tools to analyze your data. This is only about a year old. It is imperfect. I do encourage you to go check it out and let us know if it's working for you or not. Thank you. Next slide, please. And then I just say, going beyond that, trying to bring it back to our mission area in digital twins. So we think that artificial emerging technologies like AI can support real-time data capture for applications in immunology. So I think Reinhard Laubenbacher and colleagues have published this this paper on the left about utilizing digital twins in viral infection, where you could take multi-scale biological data, build an immune digital twin, and that's going to help with personalized care. And we see this as a specific use case that's relevant to the NIAID mission, where we're interested in immunology and allergic disease. Next slide, please. And I think I'll just stop there. Hopefully you can come learn more about us on the left at our data science website, where we have a blog and try to highlight papers and things of that nature. Check out the United Data Ecosystem Discovery Portal. And of course, my main point of coming here today is to alert you to the IMAG Interagency Modeling Analysis Group. So please go there. And I see others have dropped some other links in the chat. So I'll hand it back over to you. And thank you for your time.
Thank you, Reid. This was very insightful and a great overview of all the many amazing work that is going on now, that AI is part of our all lives. I think ultimately a human reference atlas cannot be created using just human intelligence alone. I think we have to bring in artificial intelligence. We have to get AI-ready data sets, and it's not easy to construct those. It's actually quite a bit of work. We have run 4 Kaggle competitions within HubMap and SendNet now, and it's a ton of work. But then you empower many to help us develop new algorithms that might actually provide the solution we need. It was interesting for me to see that all the funding agencies, every single one which presented here, they actually have collaborations with other agencies or with other institutes within NIH, and that must cause all kinds of challenges and must also be exciting, of course, at the same time. And I think we want this as researchers. We want to collaborate with the best in Europe, with the best in Asia, with the best in New Zealand. And it's not always possible. And it's in many cases very, very difficult to actually figure out how to get 2 projects funded at the very same time with the overlap it takes, so that you can really work with another team that you think is the best out there. I wonder if you could help us understand from your vantage point of what it takes and how to make that successful, so that more agencies learn about it and try it out and support that better. Because ultimately it's global science. Researchers are all around the globe. The data sets, the methods, the algorithms are around the globe. And we have to find ways to interlink them.
So it's an excellent point. I don't know, Chris or Amy, if you want to start, or I'm happy to throw out my 2 cents and then you can correct me.
Well, I mean, CIFAR is a non-traditional funder. We don't invest in the typical research grants that I think many scientists would typically apply to. But I think where we provide that opportunity for collaboration and synergy is by those small catalyst grants, again, to try to find the seed funding to be able to then feed into those future processes. But would love to hear from Reid and Chris about their interaction, I think, with other organizations. I think the other thing that we try to do too is build partnerships. And again, where are those synergies within the organization where we can hold joint calls or joint efforts to, again, further build collaborations, again, from a CIFAR's perspective in a non-traditional granting way.
Yeah, I mean, from the NIAID perspective, your points are well taken. How do you... There's a lot of different agencies out there funding different things, and then you throw in the international component, and that gets even more complicated. I'm still a recovering academic. I've been in the government about 6 years. so also was on the trying to get the funding side. I think opportunities like this are important. One way to go is really sort of like a top-down and a bottom-up approach is sort of I've seen in the last 6 years, engaging, in every agency, there's like program officials, and they're called different things in different agencies. And identifying, my advice is always to identify folks that are representing, the agencies in your interest areas and getting to know them, and getting them aware of activities that you're doing, internationally or with other agencies that we might not be aware of, and really building that relationship. And I always take like a 1 to 2 year approach on this type of thing, where, there's nothing that we can do tomorrow, but if we understand the landscape and understand, okay, NSF is doing something here, there's folks in the UK that are actually invested in, vector-borne research modeling, then what you can start to do is start to, build a relationship, formulate ideas, which then can be communicated to our leadership who are thinking about other things. That's just kind of how I've seen it, and I don't know about you, Chris.
Yeah, I would just add that I think it's important that each funding agency sort of has their own space, and you don't really want to be doing the same thing. That just creates redundancy. But there's something called the National Artificial Intelligence Research Resource that NSF is leading, but a lot of U.S. federal agencies are part of that, and it's really designed to provide large-scale computing to folks who are generating, academic AI models, who aren't going to have those resources on their own. So NSF provides, they set up the large-scale computing, and then other agencies like NIH provide the funding for the applications and the models that will be trained on those. So that's a good example. But I think it's kind of key to find that complementarity, which agency is responsible for which, and then you can partner that way.
Yeah, it's a great point. And I think also, if you take 10% of the time that you would have spent thinking about a research project and replace that with, trying to talk to program folks across these different agencies, I think that's a good return on investment, because while it's hard for me to rattle off specifics right now, we are sort of seeing these things come through our inboxes and then can communicate those back to you, and you don't have time to kind of figure out all the inner workings of the government sometimes. And so it provides you with additional eyes to see how to navigate these different areas. And again, Chris makes a very good point. Where I am, I'm focused on allergy, infectious diseases. NCI is focused on cancer, et cetera, et cetera. And so what model you're building, what's it for? That's going to sort of help you identify. You can also go to NIH Reporter and sort of put in your specific games page, and it'll pop out program officers that are like relevant to whatever you're writing, and that's a nice email. So there's lots of little tools like that.
Great. I think it's important to benefit from the complementarity and do projects together that are better than the sum of parts. And it's not easy. And within HubMap, we are dealing with 65 vital organs now, so many different NIH institutes that are responsible and caring dearly about those organs. So it has not been easy to coordinate this across. So projects like HubMap and others have been instrumental for bringing all this different expertise together. And then CIFAR now focusing on the multiscale aspect of maps and models, really, really core to have this project go forward and succeed. Thank you all for being on this panel. As you know, this is 24 hours. We are going over to hour 4 p.m. And Michael Levine just joined. I'm absolutely looking forward to Michael's presentation here. You might like to stay on, but if you can't, please do take all the things learned back to your environments. And I hope there's even more collaboration across agencies. I think this would be good for all of us researchers, which want to really collaborate with people around the globe and also collaborate across scales and across disciplines. Thank you.